So the protein or enzyme are creating from an exon, not from intron. And exons are corresponding to approximately 1% or at most 5% of the size of each gene, of the quantity of each gene. The other are intron, and they have many jobs. They are not junk DNA. Previously, some people 35, 40 years ago, they called them stupid DNA, junk DNA, garbage DNA. They insulted DNA. But after that, they all not believe God. Marxism and other ideology, they were trying to say, what you are talking is absolutely right, and the rest of the world is wrong. And they were analyzing but and now if we, even from the gene, if we delete one single basis from one intron, something must happen. Every basis, each basis on DNA, regardless is located on exon or intron, is useful, and it must be done in order to behave normally. So these are important. Okay. Now, cDNA comes from exon. I explained that easy structure of the gene in order to understand the cDNA. So genomic DNA means everything, exon and intron. But genomic cDNA means the information only from or exon. So sometimes you make cDNA because it's 1% of the genome, and much easier, much cheaper even. Why you clone the huge thing and you don't need to analyze those 95 or 99% of the information, and you want to only concentrate with the bases which are located in exon. So go to synthesize cDNA, and this is one main way, not the only way. One main way to synthesize cDNA, double-stranded cDNA. You isolate RNA, and you are sure that messenger RNA are mainly come from exons. Why I am using the word mainly, not absolutely? Because there are some exceptions that some of the information from intron also expressed in messenger RNA. Today we have this information, but they are not related to my subject today. So be careful that it doesn't mean that all information that are located in intron, they are not expressed in protein. Some of them also make proteins, especially for those genes that have several jobs. Remember at the beginning of the lecture, I said that we have some genes that have a major effect and in the same time a lots of minor effect, meaning several jobs. Those genes that have several jobs, especially because there are some, they behave, those genes during this process behave like alternative splicing, meaning like shuffling from different exons and different introns. So the combination of different exons, different introns must create different messenger RNA. And different messenger RNA must create different protein from the same sequences, because they came from different places. This is called alternative splicing. So now you realize that those people that 30, 40, 50 years ago, they called this poor intron, junk DNA, stupid DNA, garbage DNA, go away, something like that. Each individual basis has certainly one or more than one job, duty. Yeah. Okay. So if you isolate messenger RNA, so you know already messenger RNA has 150 to 200 at the preprime adenine, and polyadenylation. Sometimes U also, base U also may be there, but mainly are adenine. So if you isolate RNA for PCR, for duplication of DNA or replication of DNA, you need primer. So you make some T, make primer of, take advantage of this place, that is the primer. And also you can use of the Velar reverse transcriptase. Velar reverse transcriptase is an enzyme that first time was isolated and discovered from retroviruses. Retroviruses are the viruses that are RNA. And they have this enzyme. After that, we find that this enzyme even exists in human beings. But I'm talking about 40 years ago. Yeah, somehow, sometimes, yeah. But yeah, we do have, but not in the format that. Yeah, there is lots of debate on that, but we do. So this was discovered by Howard Tamim. Howard is a great scientist, Tamim, and David Baltimore. This gentleman also was my professor. Great man. He come to the office from other university by bicycle all the time, and he got Nobel Prize. And you know, each year there is only one Nobel Prize for medicine, not tens or 2,000. One Nobel Prize each year. And Howard Tamim was dead also. God may bless him. He was a very good man. I have lots of good memory of him, and also Watson, as I already told you. Okay, so by take advantage of this enzyme, remember, during this lecture, whenever I received you a name and that name was discovered, I respect him, I introduce him to you because this is our duty. In Farsi we say, بزرگش نخوانند اهل خرد که نام بزرگان به زشتی برد. The good man must be mentioned, must be remembered all the time, regardless from here or there. And this is our duty, to pay thanks to them, because they have done something for the welfare of the mankind. And you have to appreciate it. Not only insult everybody and things like that. It's bad to insult. Even, don't insult, please, even animals, if they are. Okay. After that, if you use of this enzyme, use that. Okay, that's DNA.
It's the time. So our next class will start at 10.
It's finished. Okay. Let me see. So I will give you this picture. Remember this and the stages and everything. And this is hard to me. I respect him again. Okay. These are other people, and these are some product of genetic engineering and biotechnology. This is Dolly, yeah, with Professor Wilwood, Jan Wilwood, with Dolly. This is Dolly and his mother.
Tamam.
This is Dolly and the mother of the Dolly. And this is a picture from that genetic engineering research center that is located between Tehran and Karaj, and I was able to establish 30 years ago.